Okay, so we need to talk about the constant region of the heavy chain protein because it's a little more complicated than I've made it out to be. So if you look on the right, there's a B cell, and on its surface is the immunoglobulin. Two heavy chains, two light chains. So the heavy chain proteins, they have a variable region and a constant region. The light chain proteins, variable region and a constant region. And so if you recall when we were talking about the heavy chain gene and the light chain genes, they're complicated because they have these gene segments that are rearranged in order to generate diversity in the variable region of the protein, giving you trillions of combinations of antigen binding sites. So if we looked at the heavy chain gene, we'd spent a lot of time talking about the variable diversity and joining gene segments, and recombination enzymes, cutting DNA. We talked about junctional diversity, putting random nucleotides between the gene segments. And so this region of the gene is going to code for the variable region of the heavy chain protein. And that makes up part of the antigen binding site. That's great. What I want to talk now is about the constant region of the heavy chain gene that codes for the constant region of the heavy chain protein. So that's all true, right? And that region that we're talking about of the protein right there is responsible for this part of the heavy chain protein here that's inserted into the membrane of the B cell. So I've been oversimplifying it because there was lots of other confusing stuff. So now I'm going to focus on the constant region because there's in fact not one constant region that is possible to be present in the heavy chain protein. There are multiple constant regions that could be chosen to be part of the heavy chain protein. So we now need to talk about the different possibilities that there are in the heavy chain constant region. So we're focusing on the constant region of the heavy chain protein. And that's going to involve the heavy chain gene having multiple pieces of DNA that could code for the constant region. So let's look at this on the next slide. So you're probably familiar with different types of antibodies: IgM, IgA, IgG, IgE, and the very little known IgD. Right? So these are all antibodies, or also known as immunoglobulins. We'll see that they can be present on the membranes of B cells. We're going to see later that they can get secreted and be used to combat an infection. But right now, let's just talk about these different antibody isotypes and their structures. Right now, they look the same, don't they? They've got two heavy chains and two light chains in terms of their proteins. But all of these immunoglobulins are slightly different. Where do they differ? They differ in the constant region of the heavy chain protein. So, for example, IgM. I just highlighted the constant region of the heavy chain protein. So the protein information in IgM, in that constant region, actually comes from a piece of DNA called constant mu. So we're using the Greek letter mu. So this protein is different, this region of the IgM heavy chain protein is different than the other ones. So, for example, IgA, its heavy chain constant region, that information is going to come from a piece of DNA called constant alpha. And so we use the Greek letter for A there, alpha. IgG, the constant region of IgG, we're going to refer to as constant gamma, so that information is going to come from a separate place. IgE, its constant region is going to be referred to as constant epsilon. And IgD, its constant region, we're going to refer to as constant delta. That's the Greek letter delta. I don't know if that's capital or small, but it's not the triangle, it's the other delta. So, all of these immunoglobulins, they could have all the same light chain, they could all have the same antigen binding site, but their heavy chain constant proteins are different, and they're going to give these antibodies different properties. So, when we talked about the structure of the heavy chain gene, we focused a lot on the VDJ region, and I just drew one constant region. But in reality, there are multiple constant regions. And so your book shows figures that display them all. And now I'm showing you the order that they're typically located in. So after the VDJ region, you have a constant mu followed by a constant delta, constant alpha, and there are multiple alphas, constant gamma, there are multiple gammas, and constant epsilons. So this is the DNA, right? This is the heavy chain gene locus. So there isn't just one piece of DNA that codes for the constant region. There are multiple pieces of DNA that code for the constant region. And you're going to have to learn how a cell and why a cell chooses different constant regions. But these are all going to give rise to these different types of immunoglobulins or antibodies. And that's good because IgG fights an infection a different way than IgA, which fights an infection a different way than IgG, or IgE or IgM. So when the cell wants to make a different antibody, we'll see how that happens in a later video. We're going to focus now just on IgM and IgD. Okay, so this is the structure, this is the more accurate structure of the heavy chain gene locus. Okay, so in a developing B cell in the bone marrow that's undergone VDJ recombination or somatic recombination, so we've already hooked up our V and D and J together, we've got junctional diversity, right? So the coding information is going to start in the variable region, and we've got all these constant regions, right? So all of them have the information to code for protein, right? And that's where the protein will terminate in the constant region. So they all have stop codons. But the cell will only choose certain constant regions to use at certain times. So I just need to get you to know when these are. When B cells are developing, and when we're talking about immature B cells and mature naive B cells, those cells typically only use the constant mu and constant delta. So they make both IgM and IgD. So let's see how that happens. So here's the DNA structure of the heavy chain gene locus. So let's say VDJ recombination has already taken place. So one of the many V's is hooked up to one of the many D's, which is hooked up to one of the many J's, junctional diversity between them. And so now this whole area is transcribed. So I drew an mRNA starting upstream at the promoter through the variable region, and you can see it went and read through both constant mu and constant delta. So we've got this we have this piece of RNA in yellow. Now you hopefully remember splicing. If you learned it in genetics or molecular biology or biochemistry, splicing is going to go on here. That's going to allow us to make two different proteins. So this region of the mRNA, that will code for the variable region of the heavy chain protein. These other regions here will code for the constant region of the heavy chain protein. So what happens in a B cell is alternative splicing. And if you remember what alternative splicing is, usually you think about it in terms of introns and exons. Now we're thinking about it in terms of different constant regions of the heavy chain protein. So in a B cell that expresses both IgM and IgM and IgD, what happens is there is some splicing that just hooks up the variable region with the constant mu region. And so that's going to give you a piece of mRNA that looks like this. And that's going to get translated into protein that will give you IgM. Some of the RNA, it is spliced differently. So when it is spliced, it hooks up the variable region to the constant delta segment of mRNA. And that gives you a different piece of mRNA that's mature. So when these go out into the cytoplasm and find ribosomes, they're going to be translated, and so the one with constant mu gets translated into protein, heavy chain protein, that's going to give you IgM. The one with constant delta is going to translate to give you protein that will give you IgD. They both have exactly the same variable region, which means they're going to have exactly the same antigen binding site, but they have different constant regions. So if we look at this B cell here, on its surface, what are we going to find? We find IgM on its surface, and we also find IgD on its surface. And most mature naive B cells that are circulating through your body right now express both IgM and IgD membrane bound, and that's important as well. So, again, how does a cell do this? Well, it's got this heavy chain gene where it can choose and use multiple constant regions. So these two immunoglobulins, they have the exact same antigen binding site. And again, antigen binding sites are generated because of the variable region of the heavy chain and light chain gene. So these IgM and IgD, they use the exact same variable regions. What they're using are different constant regions. So this is our first introduction into the fact that immunoglobulins, specifically the heavy chain protein, can have different constant regions. So IgM and IgD are the first ones we talk about because those are present in the developing B cell in the bone marrow and the mature naive B cell that circulates through the body. When B cells become activated, only then will switching occur to the other constant regions that could give you IgA or IgG or IgE. That will be for another video.